Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Accelerating Vaccines to Clinical Trials, a Toolkit for Efficacy, Safety, and Bioanalytical Studies. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Aerogen Life Sciences who developed the content for this presentation. Aerogen Life Sciences is a leading R&D manufacturing solutions provider for the life sciences industries worldwide. It offers end-to-end integrated or standalone solutions for small and large molecules. Established in 2001, the company operates through a network of sites located in India and Morgan Hill, California, USA, with a team of 3,700 plus scientists and more than 450 PhDs. Its expertise and experience have enabled over 400 customers to advance their research programs from discovery through commercialization. Intox Private Limited, a subsidiary of Aerogen Life Sciences, is an OECD GLP certified preclinical safety assessment service provider with over 25 years of expertise in conducting safety assessment studies for pharmaceuticals, biochemicals, biopharmaceuticals, medical devices, agrochemicals, industrial chemicals, nutraceuticals, dietary supplements, cosmetics, and others. Now it's my pleasure to introduce you to our speakers for today's webinar. And first, I'd like to introduce you to Dr. Narendra Deshmukh. He's Executive Director of Intox Private Limited. Narendra there he is. Hi, Narendra. He is one of the co-founders of Intox Private Limited, a wholly owned subsidiary of Aerogen. He has over 3 decades of experience in regulatory toxicology and toxicologic pathology and has executed several safety assessment studies for a diverse range of products, including chemicals, pharmaceuticals, vaccines, crop protection, and medical devices. He is the former director and charter diplomat of the Indian Board of Toxicologic Pathology, diplomat of the American Board of Toxicology, and president of the Society of Toxicologic Pathology in India. Now it's my pleasure to introduce you to your second speaker, Dr. Kedar Pranapatri, Director of Bioanalytical and Bioassays at Intox Private Limited, a subsidiary of Aerogen Life Sciences, and there he is. Hi, Kedar. Kedar holds a PhD in molecular biology from the Indian Institute of Sciences. He has over 40 publications and 2 patents. Kedar has served as a visiting editor for journals like Cancer Research, Drug Design Development, and Therapy, and International Journal of High Throughput Screening. He has over 22 years of experience in the pharmaceutical industry and contract research organizations, specializing in bioanalysis of small and large molecule therapeutics. Now it's my pleasure to pass over the mic to our first speaker, Narendra. So Narendra, when you're ready, you may begin.
Thank you, Sonia, for the wonderful introduction. Hello, everyone. We are very happy to welcome you to our webinar on accelerating vaccines to clinical trials, a toolkit for efficacy, safety, and bioanalytical studies. In this webinar, we are going to have a brief overview of the vaccines, their changing platforms, and the concerns that change with them. We would look at the regulatory pathways for vaccines, the time involved, and would try to identify the means to accelerate these pathways to take us faster to the stage of starting the clinical trials. While emphasizing on the non-clinical research and development, we would touch upon the importance of conducting the efficacy and safety studies in an appropriate and efficient manner so as to accelerate your vaccine development programs to the filing of an investigational new drug application. To highlight some of the key requirements, we would also share with you some examples from the real studies that we have conducted at Intox Arajan. Next slide, please. Let us begin with the definition of the vaccine as cited in the latest guideline on clinical evaluation of vaccines by the European Medicines Agency in January 2023. Vaccines are defined as medicinal products intended for prevention, post-exposure prophylaxis, and or treatment of diseases caused by an infectious agent and which contain antigen or genetic information for an antigen, either of biological or synthetic nature, that induce a specific immune response against the causative infectious agent or its toxin. History of vaccines spans over centuries, preventing smallpox, rabies, foulpox, diphtheria, pertussis, influenza, yellow fever, polio, and hepatitis B. These early traditional vaccines focused on prevention of infectious diseases, eliciting humoral immune responses. These traditional vaccines are typically composed of whole, inactivated, or attenuated microorganisms, which can be bacteria or viruses that have lost their disease producing properties. However, the next generation vaccines have been designed not only for prevention of infectious diseases, but also for treating chronic diseases such as hepatitis C or cancer. These next generation vaccines aim to induce strong humoral and cell mediated immune responses and include both prophylactic and therapeutic vaccines. However, for the purpose of the present webinar, we shall be restricting our discussions to the prophylactic vaccines and not to the therapeutic ones. Next slide, please. As depicted in this cartoon, in last 25 years at Intox Arajan, we have tested several traditional vaccines as well as the next generation vaccines. The recent pandemic offered us several insights in testing the next generation vaccines like virus vectored vaccines, DNA vaccines, and mRNA vaccines. Besides several toxoid vaccines, we also tested COVID-19 vaccines made from virus-like particles and several subunit vaccines, including polysaccharide conjugate vaccines. The era of whole organism vaccines is not yet over. In last some years, we have tested both inactivated as well as live attenuated vaccines such as the H1N1 influenza vaccines. And very recently, we completed a study on a Leishmania vaccine, which is a live parasite, not capable to replicate and is not pathogenic. So as the vaccines evolved over last several years and the platforms changed, these have come up with new and newer challenges and concerns. And with that, the regulatory process to develop and take these vaccines to the clinics has also become a lot more complex and time consuming. If we take a look at the regulatory pathway for vaccines, we realize that after the initial exploratory period of 2 to 5 years, the evaluation of preclinical efficacy and safety can take about 2 years, and thereafter, upon obtaining the permission to start clinical trials, the phase 1, phase 2, and phase 3 of the clinical trials can respectively take 1 to 2 years, 2 to 3 years, and 2 to 4 years. And finally, the regulatory process of reviewing all the preclinical and clinical data takes about 1 to 2 years. Thereby, the entire process of taking the vaccines to the clinics can take as long as 10 to 15 years. However, we also are aware that during the pandemic, under emergency use authorization, this process was completed within about a year and a half. We need to figure out what was done differently when these timelines were compressed during the pandemic to a shorter period of about a year and a half. We also need to find out whether the 2 years period required for non-clinical efficacy and safety research can be reduced and how. So if we look closely to the key components of accelerated vaccine development during the period of pandemic, we realize that the entire process was compressed in time by application of extensive current knowledge. Also, more resources were mobilized simultaneously, and the efforts were supported by early and continuous dialogue between the developers and dedicated group of regulatory experts and regulatory agencies. Besides, the companies also emphasized on expanding their manufacturing and production capacity concurrently. So, since simultaneous reviews by regulatory agencies and emergency use authorization can happen only under the situations of a pandemic and are beyond our control, under normal circumstances, to accelerate the vaccines to clinical trials, the focus must be on speeding up the early research and the non-clinical development. We believe that this can be achieved by choosing the right tools and models for the evaluation of the efficacy and safety of the vaccine candidate. A thorough understanding of the mechanism of action of the candidate vaccine will be required to select methods that are appropriate, efficient, and acceptable to the regulatory agencies. A good understanding of the concerns around the vaccine, its ingredients, its method of administration, and of the regulatory test guidelines for addressing such concerns all will be required to design, plan, and perform GLP compliant non-clinical safety assessment program in a speedy manner. I request Dr. Kedar to provide you a glimpse of the tools and methods that can be employed to ensure that the efficacy studies are appropriate and efficient in meeting the regulatory requirements for immunogenicity and the protective properties of a candidate vaccine.
Thank you, Dr. Deshmukh. To accelerate your vaccine development program, it is crucial to understand the mechanism of action of the test vaccine. The immune response to vaccine is driven by both T and B cells. The adaptive immune response begins in the lymph nodes where the dendritic cells, also referred as antigen-presenting cells, process the vaccine and present vaccine antigens to T lymphocytes and B lymphocytes. Bacteria and viral antigens employ distinct pathways to evolve these immune responses. Bacterial or parasitic antigens trigger antibody-mediated immunity by binding to MHC2 class of proteins and interacting with CD4 cells, while viral antigens trigger cell-mediated immunity by binding to MHC1 class of proteins and interacting with CD8 cells. The efficacy markers differs for each class of pathogens, and understanding the appropriate biomarkers is very critical for designing vaccine efficacy studies. Initial immune markers in the efficacy studies antibody titers against the vaccine antigens. As vaccines are expected to offer protection against multiple variants of antigens, hence to expedite your program, it is advisable to measure the antibody titers against multiple variants. At Intox Aragen, we use validated assays on mesoscale discovery platform, which allows us to test at least antibody titers against 10 different antigens in a single well using minimal anti-sera. Regulators also expect to provide data for functional antibody titers. Examples include opsonophagocytic activity and serum bactericidal activity, which I have shown on this slide. Both the assays rely on the principle that antigen-specific antibody coat on the target pathogen in presence of complement, and this process is called as opsonization. The endpoints of both the assays are different. In serum bactericidal activity, the complement activation pathway creates the membrane-associated complexes, which create pores inside the cells and lead to the bactericidal activity. While in OPA assay, phagocytes are provided in trans and they phagocytize opsonize antibodies. The endpoint is the number of bacteria that are surviving. This is used to assess the efficacy of vaccine. I have here two examples. The vaccine for serum bactericidal activity, it's a classical assay for meningococcal vaccine program. And recently, the CDCO side recommends that the vaccine efficacy can be estimated based on the serum bacterial titers of the vaccine in absence of direct evidence of clinical effectiveness. For pneumococcal conjugate vaccine, the Prevnar is being taken from 13 valent to 20 valent, and an accelerated approval was for this 20 valent vaccine was done based on the OPA activity results for each added strain. Regulators emphasize on use of suitable and functional antibody assays to accelerate approval of your vaccine development program. Critical requirement for any vaccine is the potential to elicit memory. T-cell mediated memory is assessed by administering repeated vaccine doses, collecting the spleen samples, and then co-incubating them with the vaccine peptides, positive control concavalin A, or media control. And the biomarkers, the endpoints are cytokines, T-cell activation markers, and T-cell proliferation. In this slide, I have also shared the data that were generated in-house with the multivalent dengue vaccine, and we could clearly demonstrate that the vaccine was active against different variants using such assay. The pivotal study in vaccine efficacy study is to demonstrate the protective effect of vaccine. These are done by repeatedly administering the vaccines and challenging them with the lethal doses of target pathogens. The in vivo protective properties of vaccines are evaluated through comparison of body weights, survival post-infection, demonstration of the target antigens by qPCR or through immunohistochemistry. Accelerating vaccine program can involve creating new models. To perform such efficacy studies, these are done through two major approaches. The classical approach is to passage the vaccine through the same species that is available in the lab. Here is an example where SARS-CoV-2 was passaged to the immunocompetent mice, and upon sixth passage, a strain was identified which could create symptoms like human diseases. And this mouse had a mutation from asparagine 501 to tyrosine. Another approach that is being used is to create transgenic animals. And this is an example, a study done at Chulalongkorn University, where the transgenic animal was administered with multiple doses of vaccine and then challenged with SARS-CoV-2 by intranasal route. The group has shown that the vaccine titers, the pathogen titers, were highly reduced in the vaccinated animals compared to the control animals. Another way to accelerate your vaccine program is to use new vectors. As we see risk of vector-specific antibodies for existing vectors. Lentiviruses are a challenge, are being used or are being an attractive vector for the research, as they have very good potential or very high potential to directly transduce into antigen-presenting dendritic cells, and also they have low pro-inflammatory risks. However, FDA recommends meticulous preclinical toxicological evaluation to dial out toxicities due to residual host cell protein, residual nucleases, bovine serum albumin, and residual endotoxins. These studies are have to be done very carefully, and it requires a highly specialized CRO that can afford such services. All our vaccine efficacy studies at Intox are supported by a highly experienced bioanalytical team. It's in a dedicated GLP compliant biocell laboratory equipped with sensitive and calibrated equipments. I request Dr. Deshmukh to steer the discussion through the intricate aspects of the non-clinical safety assessment.
Yeah, thank you, Dr. Kedar. As said previously, a good understanding of the concerns around a candidate vaccine, its ingredients, its method of administration, and of the regulatory test guidelines that are addressing such concerns, all will be required to design, plan, and perform GLP compliant non-clinical safety assessment program in a speedy manner. Historically, the vaccines were considered as safe. The traditional vaccines were usually tested for their efficacy only, and there was not much emphasis on conduct of toxicity studies. But over time, the extent of non-clinical safety testing has been greatly increased, and requirement for full toxicology have been laid down in the regulations. There are two primary safety concerns around the vaccine: local reactions and systemic reactions. Please go back to the previous slide, Dr. Kedar, if you can. Local reactions are often observed with intramuscular or subcutaneous routes of administration. They manifest as pain, redness, and swelling, and often there could be formation of granulomas and abscesses at the injection sites, along with necrosis and regional lymphadenopathy. As the vaccine platform technologies evolved to the modern next generation platforms, the safety concerns around the vaccines have also evolved. Potentially severe systemic responses of traditional vaccines include anaphylaxis, fever, organ-specific toxicity, or immune-mediated toxicities such as cytokine release, immune activation, or suppression and autoimmune diseases, and effects on reproduction and development. But with advent of next generation adjuvanted vaccines, there have been additional concerns on the potential synergies and interactions between the mechanism of action for vaccine antigens and the adjuvants. There have been concerns that the adjuvants would enhance the immune response and possibly cause excessive amounts of pro-inflammatory and pyrogenic mediators, which could ultimately lead to exacerbation of both local and systemic effects. As the new generation platforms such as viral vectored vaccines, DNA vaccines, and mRNA vaccines evolved, these raised additional concerns, and then the regulations were set around addressing such concerns. However, over the time, as more studies were conducted and published, more data became available, and regulatory agencies accumulated a wealth of knowledge about these platform technologies. Their concerns have also changed, and this was reflected in the revisions of testing guidelines. It would be interesting to see how the concerns like integration, biodistribution, autoimmunity, and immunopathology are no more considered as risks for DNA vaccines, unlike they were considered in 2005. Similarly, integration with the host genome is no more considered a risk for mRNA vaccines. The current concerns for mRNA vaccines have been listed in the 2021 guideline for WHO for mRNA vaccines, and they are towards biodistribution and persistence. Then they are for inflammation caused by the mRNA vaccines, the unexpected and serious toxicities from modified nucleosides, and adverse effects exerted by novel lipids, novel lipid nanoparticles, and novel formulations. In fact, for such platforms, the delivery system has also become more relevant than the antigens themselves. The recent WHO guidance for DNA vaccines of 2021 reads that based on clinical experience, the observed reactogenicity to DNA vaccines appears to relate more to their delivery method than to the DNA vaccine itself, and most notably in the case of electroporation or particle mediated bombardment. Hence, it becomes really important that the toxicologists engaged into designing, planning, and conduct of non-clinical safety studies on vaccines make themselves adept and familiar with ever-changing scenarios of regulatory guidelines. The 2005 guideline of WHO which is applicable to all vaccines, and the subsequent 2013 guideline applicable to adjuvants and adjuvanted vaccines can be considered to be the gold standards when it comes to non-clinical safety assessment of vaccines. Of course, besides WHO, there are several other respective national regulations like those by US FDA, CBER, by the European Medicines Agency. Then in Asian scenario, we have the Japanese, the Chinese, and Indian regulatory agencies. And all these guidelines need to be referred by the toxicologist before planning a toxicology program for a candidate vaccine. So these test guidelines describe various aspects of the design of a non-clinical safety assessment program and also describe the finer aspects of the experimental protocols, such as choice of studies, choice of relevant animal species, duration of the study, number and frequency of doses, route of administration, groups to be tested, and the various parameters that need to be evaluated in such studies. It is therefore critical for toxicologists aiming at speeding up of the non-clinical research and development of a vaccine that he or she does not miss the testing requirements that are unique for your vaccine candidate. Repeated dose toxicity study is considered as the pivotal toxicology study that is performed prior to the clinical trials. Although there are single dose toxicity studies and local tolerance studies described in the requirements, it is understood and accepted that these studies need not be conducted as standalone studies, but they can be incorporated in the experimental designs of the repeated dose toxicity study. However, when it comes to viral vectored vaccines, there is a possibility of a pre-existing vector immunity in the test animals, and it might interfere with the response to a subsequent dose for the test vaccine, which can potentially compromise the results of a repeated dose toxicity study. In such cases, a single dose toxicity study may suffice, provided that the intended human schedule is also a single dose. The developmental and reproductive toxicity studies, commonly known as DART, are required if the vaccine is indicated for pregnant women or women of childbearing potential. For this study, a prenatal and postnatal developmental toxicity study is generally considered to be adequate. This study focuses on female fertility, teratogenicity, and postnatal effects, which are the main concerns for reproductive toxicity studies on vaccines. If in the early clinical trials there are going to be appropriate measures taken to avoid pregnancy, then even this reproduction toxicity study can be delayed or deferred to a later stage, but before the marketing authorization application can be issued. When it comes to evaluation of effects on male reproductive system, the data generated during the repeated dose toxicity study are usually considered to be adequate. Genetic toxicity studies and carcinogenicity studies are not normally required for final vaccine formulation. However, a standard battery of genotoxicity studies is generally recommended for most novel adjuvants that contain new chemical entities. The biodistribution, next slide. The biodistribution and persistence studies are required for mRNA vaccines and viral vector vaccines. When conducted as standalone studies, these would reveal the kinetics to a greater detail. But the biodistribution studies are also incorporated in the repeat dose toxicity protocols, and these assessments are made at termination of the experimental animals. Pharmacokinetics and toxicokinetic studies are not normally required unless there are specific concerns due to an adjuvant. Similarly, safety pharmacology studies are not normally required for vaccines. However, if during the repeat dose non-clinical tox study or during any clinical studies, there are reasons to raise any concerns for the effects on cardiovascular, respiratory, and central nervous systems, then specific studies could be asked by the regulatory agencies to be conducted. Certain endpoints of safety pharmacological studies can also be incorporated in the protocols of a repeat dose toxicity study. Let us look at the choice of animal species. And when it comes to the choice of the animal species for a repeat dose toxicity study, conducting a single study in one biologically relevant species is considered as sufficient. A biologically or pharmacologically relevant species would be one that would demonstrate immunogenicity against a vaccine antigen. For adjuvanted vaccines, it is also a desire that the adjuvant enhances the immune response in the animal species by the same mechanism as it does in humans. Rodents and rabbits are the most commonly employed animal species, while non-human primates are only used if no other relevant species exist. It is highly recommended that the animal species chosen is one for which relevant and sufficient historical control data exist in the test facility. At Intox Aragen, we have excellent historical control data, which really helps us analyze and interpret the data from toxicity studies. Uh, a second species would be required to be tested only in special circumstances where it is necessary to further characterize the candidate vaccine. These testing guidelines have also specified the numbers of animals that should be included in the main toxicity groups and the recovery groups. The route of administration of the vaccine has to be the same as that will be used in the clinical trials. Most commonly, the routes are intramuscular, subcutaneous, or intradermal. Also, if the clinical trials are going to use any specific device for dose administration, then it is expected that the same device be employed in the animal toxicity studies. At Intox Aragen, we have experience of testing a novel COVID-19 vaccine, which was intended to be injected to human subjects by means of a needle-free intradermal injection device. So while conducting the animal tox study, we ensured that the same device was procured in our test facility and it was employed for giving injection to the test animals. The dose that maximizes exposure of the animals to the candidate vaccine and also induces peak antibody response is expected to be tested in the animal toxicity studies. Normally, a vaccine is required to be tested at only one dose, and this is the intended maximum clinical dose in absolute terms. For example, if the human dose is going to be half ml, then the animals will receive the same half ml dose, and the dose will not be based upon per kg body weight basis. If, due to the reasons of animal welfare, such larger volumes of dose cannot be injected to the animals, and we often find it in case of intramuscular injections, then the dose volume can be split and injected at more than one sites to the animal. And if even this is not possible, then only a dose that exceeds the human dose on a mg per kg body weight basis, but is capable of inducing immune response, should be tested in the animals. It should be noted that unlike the other toxicity studies, other small molecules and other pharmaceuticals, low and mid doses are not required to be tested because identifying a lethal dose or dose levels such as NOEL or NOAEL is not the objective of a vaccine toxicity study. Dose response is not required to be demonstrated in these studies. However, when the vaccine contains a novel adjuvant for which there is no previous safety data available, then only it is expected to test more than one doses of the adjuvanted vaccine to identify a dose response relationship for the adjuvant. In a repeated dose toxicity study, the number of doses to be tested are required to be equal or more than the number of doses intended to be administered in the clinical trial. Most commonly, one additional dose is chosen, and this is referred to as N plus 1. In a vaccine study, the doses are not to be injected every day. It is expected that the interval between the doses in animal toxicity studies can be shorter than that in the proposed clinical trial. So typically the intervals can be 2 to 3 weeks. The test guidelines also require a recovery period, the period after the last dose when the animals are simply kept under observations and then terminated. The recovery period can be of 2 to 4 weeks, depending upon any expected effects or the possibility of delayed toxicity. As discussed previously, in repeat dose toxicity study, normally a vaccine is required to be tested at only one dose, and this is the intended maximum clinical dose in absolute terms. Besides, it is expected to have appropriate concurrent control groups in the study. Such groups can include a negative control group, or if appropriate, an active control group which would contain the vaccine formulation without an adjuvant, without an antigen. For adjuvanted vaccines, when the adjuvant is likely to induce any effects of its own, an adjuvant control group proves to be helpful to interpret the observed effects. Occasionally, a national regulatory agency may ask a sponsor to include a reference vaccine in the experimental design to demonstrate and compare safety of the novel vaccine against an approved vaccine that is already in the market. This table is a representative study design for an adjuvanted vaccine on which a 4-week repeated dose toxicity study with 2 weeks recovery period will be conducted. So you can see one negative control group, one adjuvant control group, and a group dosed with the adjuvanted test vaccine. As we discussed previously, you can see that a concurrent cohort of rats has been included for assessment of acute toxicity following a single dose of the vaccine. These rats will receive a single dose on day 1, and then they will be terminated after 48 hours, that is on day 3, and will be subjected to laboratory investigations and necropsy, followed by histopathology of the injection site. Likewise, you can also see a concurrent recovery group of rats included for each of the study groups. In this 4-week study, 3 injections will be made on day 1, day 15, and on day 29 with an interval of 2 weeks. And the rats will be terminated 2 days after receiving the last dose of the vaccine, that is on day 31. The recovery groups will continue to be observed for a period of 2 more weeks and then will be terminated on day 43. Clinical pathology assessments, including determination of acute phase response, will be made pre-dose and at termination of the rats. Terminal evaluations will include necropsy, organ weights, and histopathology. It should be noted that if biodistribution is to be assessed for any vaccine, then we will have to include an additional cohort of rats for this purpose. Let's have a look at the test parameters in the repeat dose toxicity studies. These include in-life observations such as survival, clinical signs, ophthalmoscopy, body weight, food consumption, and body temperatures. Recording body temperatures is considered important for adjuvanted vaccines, and these are recorded before making the injection and thereafter at 4 to 6 hours and at 24 hours after the injection. And if there is a rise in temperatures, then the observations are continued until the temperatures return to the normal. A careful examination of the injection sites is of paramount importance for studies on vaccines, and it is advised to assign grades or DRE scores to describe the reaction. Laboratory investigations comprise of clinical hematology, coagulation parameters, serum chemistry, and occasional urine analysis. The laboratory investigations are conducted pre-dose and at about 1 to 3 days following the administration of the first and the last dose of the vaccine. The immune responses are evaluated by measuring the antibody response and in some cases by further determining the immunoglobulin classes. These evaluations are conducted pre-dose and at termination of the animals. However, they may be additionally conducted midway during the study also. Acute phase reactants are also measured pre-dose, then midway during the study to reflect the peak elevation, and at termination of the animals. It is important to note that these acute phase reactants are species specific. For example, alpha 2 macroglobulin is specific for rats, while C reactive protein is specific for rabbits. At termination of the treatment of the or the recovery periods, the animals are subjected to a gross necropsy and their organ weights are recorded. Tissues are collected for histopathology as well as for biodistribution studies from satellite group of animals. It is important to ensure that the bioanalytical methods are validated under GMP. Histopathology evaluations focus on pivotal organs such as brain, lungs, heart, kidneys, liver, and reproductive organs. It is important to collect tissue specimen from the site of vaccine administration for the microscopic examination, and histopathology of organs of the immune system is considered critical, and it is imperative to collect and examine organs like lymph nodes, which are like the draining lymph nodes at the injection sites and also those that are distant from the injection site. They also include thymus, spleen, bone marrow, Peyer's patches, or bronchus-associated lymphoid tissues. With this, we would like you to further take through these evaluations by citing case studies from our own test facility. So this case study that I'm presenting it was a prenatal and postnatal developmental toxicity study in rats on a DNA COVID vaccine. In such study, the females are required to be treated with the vaccine well in advance before they are mated, which ensures that peak antibody response is present during the period of organogenesis. Therefore, in the present study, we made a single injection with the vaccine at 3 weeks before the females were cohabited with the males. Further, during the gestation, administration of one or several additional doses is recommended during the period of organogenesis, which for rats is the period between implantation to closure of the hot palate. Such exposures allow for evaluation of the potential direct embryotoxic effects of the components of the vaccine formulation. And also, depending on the animal model, it allows the maternal antibody to transfer to the progeny during pregnancy or during the lactation period. In this study, we administered 2 such doses to the pregnant females on gestation day 6 and day 15. All the pregnant dams were then divided into 2 subgroups or 2 arms. So about half of the dams were subjected to cesarean section on day 20 of gestation, which was 1 day prior to their scheduled day of parturition. These dams were subjected to examination of their ovaries and uterine contents. Their fetuses were removed and subjected to external examinations and subsequently to visceral and skeletal examinations for detection of any structural abnormalities. But the other subgroup of dams was, however, allowed to litter normally and allowed to rear their offspring through the lactation period of 3 weeks. These dams received an additional 4th injection of the vaccine on 7th day of their lactation period. At end of 3 weeks of this period, the dams were terminated along with their pups and then were subjected to various evaluations. I request Dr. Kedar to explain to you the immunogenicity and biodistribution assessments that were performed on the dams and pups in this study.
Thank you, Dr. Deshmukh. So in this study, we have evaluated the immunogenicity and biodistribution of vaccine plasmid in all groups of animals, both cesarean section as well as weaning group. We observed antibody titers in all study groups. The titers in arm C fetuses were due to the placental transfer of antibodies. Interestingly, the titers in the 21-day-old pups were due to the transfer of antibodies from mother's milk. This data demonstrates that maternal vaccination of SARS-CoV-2 mRNA offers protection to the suckling pups. Regarding the biodistribution of plasmid DNA, regulators had kept it optional as the data for biodistribution of plasmid DNA was available in adult animals. However, the agencies urged industry to provide such data for infants. We discussed this with the study sponsor and suggested to include the fetuses as well in the study of vaccine DNA biodistribution. The vaccine DNA was analyzed from 11 tissues from both dams and fetuses from both arm C and arm W groups using an in-house validated quantitative PCR assay. We collected tissues for all groups 48 hours after the last dose and analyzed for vaccine DNA. No plasmid vaccine DNA was detected 40 hours post last dose, indicating that plasmid DNA was not persistent in these animals. With this, we have demonstrated that plasmid DNA is not persistent and the DNA vaccine provides protection through generation of specific antibodies in pregnant females. In another case study evaluating mRNA vaccine in rats, we have looked at the inflammatory response and biodistribution and immunogenicity. Inflammatory response can be generated by the vaccine mRNA as well as the adjuvant used in the formulation. Repeated doses of 25 microgram of SARS-CoV-2 mRNA was administered by intramuscular route, and another group was treated with 10 microgram of SARS-CoV-2 mRNA. The inflammatory biomarker alpha-2 macroglobulin levels were tested 48 hours after each dose administration. The alpha-2 microglobulin response was induced when the vaccine was administered by intramuscular route. However, we did not detect any levels above the background when it was administered via the intradermal route. Both routes generated antibodies against the target proteins in rat. Further, in the same model, we have provided an extensive biodistribution study using 11 tissues after first dosing, using and validated multiplex RT-qPCR reaction with vaccine-specific probes and primers. The peak concentration of viral RNA were detected in lymph nodes, which is the primary site of antigen presentation to generate immune response within 6 hours of administration of vaccine. We also analyzed RNA from 11 tissues 48 hours post-administration to identify if there is any persistence with this mRNA. Interestingly, we could not detect any mRNA concentrations in all the tissues that were tested after the last dose, indicating that there is no persistence of vaccine mRNA. So we saw a limited biodistribution only to blood and injection site and lymph nodes, suggesting that a vaccine does not cause any other non-specific toxicity. At Aragen, we tailor our vaccine safety efficacy packages to meet our clients' specific needs. In a recent COVID mRNA vaccine study, our client required complete safety data within 6 months to proceed with the scheduled clinical trial. We achieved these goals through meticulous planning, simultaneous development of in-house sensitive methods, simultaneous conduct of method validation and in-life phases, simultaneous analysis of clinical chemistry, immunogenicity, biodistribution, and histopathology phases. An interim report was provided for the review of the Clinical Ethics Committee. Our efforts were in full compliance with GLP standards with efficient quality assurance unit audits. The comprehensive reports we submitted facilitated our clients' smooth progression into clinical trials. In summary, our current offerings for GLP complementary safety assessment of vaccines are designed to provide complete safety data within 32 weeks time frame, supporting your IND filings and beyond. Thank you for your time, and now we are open for questions from the audience.
Well, firstly, I'd like to say thank you very much, Dr. Narendra and Dr. Kadar, for that very insightful presentation. And we are going to begin the Q&A portion of the webinar. So I'll invite our speakers to put on their webcam. Hi, everyone. And we will begin. Just as a reminder to our audience, you still can send in your questions right now by using that questions window, and we'll try to attend to your questions during the time that we have together. So let's start with this first question here. This audience member is asking, which is the preferred species for safety assessment of vaccines? Who would like to start with that? Dr. Kadar or Dr.
Uh,
Narendra?
yeah,
Deshmukh, yeah.
I'll take up this
Okay, thank you.
question. So, well, actually, I dealt with this question elaborately during the presentation. But the most important attribute to select a species that it should be biologically relevant, so that it should be eliciting immune response to the vaccine in question. It is also expected by some agencies that the choice of the species should be also governed by the similarity or the similarity of the disease, that the animal species and the humans, the disease should be similar in expression. But
Mhm.
actually, this is not always possible. So we can just settle with the basic requirement of having an immunogenic potential for the vaccine. I would like to add that the vaccines normally act in various ways, more than one ways in the body, in
Mhm.
human subjects. And while we do a non-clinical safety study, it is not always essential that the animal model also expresses the response to the vaccine in exactly the same manner as that would happen in human subjects. So that can really make the toxicologist happy, because we have very limited choice. The second most important attribute I would say is availability of historical control data, which is very, very important, because you come across a lot of variations in the sample size in a toxicity study are pretty small. I mean, although we use 5 or 10 animals, they are still small. So you have to compare the findings with the concurrent control, but also you have to refer to your historical control data. So I think these two important attributes should govern the choice of the animal species. Thank you. I hope I answered it.
Okay, thank you very much for being so detailed. Dr. Kadar, did you want to add anything to that before I go to the next question?
I think he has covered all the aspects. So yeah, you can go to the
Okay.
next one.
Okay, thank you. All right, here's our next question. This audience member is asking, what about the biodistribution of mRNA vaccines in ovaries and other reproductive organs?
Yes, we have tested it, and we did not detect any mRNA in both organs. So mRNA vaccines are safe; they cannot be transferred from a generation to other. Yep. We
Okay,
could only
thank you.
see them in the three tissues that I had shown. They were in blood. We saw it in lymph node and some part of RNA in the injection site.
Okay, all right, thank you very much. We'll go to our next question. As a reminder to our audience, if we run out of time, don't worry, we will get to your questions after the presentation. But if you still have questions, please go ahead and send them in. This audience, the same audience member that sent that first question, he has an add-on to it. He's saying, or he or she are saying, and the difference between the mRNA and the liposome carrier.
We have looked only at the mRNA by RT-qPCR. We have not monitored the liposome carrier in the biodistribution study that was done in-house.
Okay, all right. Thank you very much for that, Dr. Kadar. Okay, we'll go to our next question, and this looks like it is the final question. What are the key differences in safety assessments of any mAbs and vaccines? Who would like to start with that?
Again, Dr. Deshmukh, it's his domain.
Okay.
I can
I
talk
think
about the bioanalysis.
this
Yeah.
is a this is a funny question, very interesting.
Mhm.
These are completely two different kind of therapeutics. The purposes are different, the targets are different, and the purposes of safety assessments are slightly different. Vaccines are meant to elicit an immunogenic response, whereas when we are going to test MAPS, we fear the immunogenicity response, because they are going to come into the way of action of those monoclonal antibodies.
Mm.
Um, Another key difference could be, of course, the choice of species. See, again, when you test the monoclonal antibody therapeutics, it is important that you test it into a relevant animal species. And here the relevant animal species means that particular therapeutic antibody should be able to act, or it should have the appropriate receptors in that animal species. So whereas in case of vaccines, we say that it should be immunogenic. And most often when we do studies on in laboratory animals and with a vaccine, the antigens are usually allogenic. They are foreign, so eliciting antibody response is very much expected. Thirdly, I would say while testing a monoclonal antibody, we have to test low, mid, and high doses. We have to establish a dose-response relationship, and we have to arrive on a no observed adverse effect level. So that is going to be used as a basis for the first human dose in the clinical trial.
Okay, thank you very much.
Coming from bioanalytical part, I think testing monoclonal antibodies is relatively easy because you have only one antigen. But with vaccines, you have to deal with a number of antigens. So vaccine bioanalytical is more challenging than doing it for monoclonal antibodies.
Yeah, I think one more difference, Dr. Kedar, would be we are not interested in looking at the kinetics, pharmacokinetics, or toxicokinetics the way we look for therapeutic monoclonal antibodies. We don't go for that way in case of vaccines.
Absolutely. Right.
Okay, well, thank you very much, gentlemen. Well, thank you very much for those questions and for answering. We have reached the end of the Q&A portion of this webinar, and if we couldn't attend to your questions, the team at Aerogen Life Sciences will follow up with you after this presentation. And if you have any further questions, please utilize the email address that you see there on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive of this event. Additionally, there is a link to view the recording of this event, which you can also share with your colleagues so they may register for the recording here as well. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. So I encourage you to do that. Now, please join us in thanking our speakers, Kedar and Narendra, for that very insightful presentation and for answering all your questions. So thank you very much. And there they are.
Right.
Thank you.
you.
We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until next time, I'm Sonia Hunt. Please take care and bye for now. Bye, everyone.
Right.
Yeah, bye-bye. Thank you, Sonia. Bye-bye.
Thank you.